So welcome back on my other lessons about biology. So this whole unit, we're going to be talking about the biology content we learned in Science 10. So let's get to it. There'll be around 8 parts and many different presentations will be cut into half depending on their size. So the first one we're going to talk, oh shit, I need to shave. Okay, the first one we're going to be talking about is history and life. So what is life? And the answer is not ball. That's what I think every single time. Living things, they all share 5 characteristics. They all require energy to grow. That's important. All living things require energy. All living things produce waste. All living things respond to different stimuli in their environment. If you can guess the next one, it'll be all living things interact. Oh no, they reproduce. All living things reproduce and they grow. So to summarize that, all living things require energy. Why? Because energy allows them to perform their biological processes and functions. They need to produce waste because of every process and every function that happens. There's always a byproduct that needs to be removed. They respond to stimuli, so they interact with their environment and the different things around their environment. Oh wow, look at that. um, they reproduce so that life can continue and life can grow on, and they grow so that they can better adapt themselves. Now let's start off by talking about some cell stories. Technology in our modern environment leads to better observations. When we have better microscopes, we can see things much more clearly. So back then, technology wasn't as modern, but as soon as technology started developing, cellular research and biology as a subset of science started developing also. So, like I just said, microscope and imaging technologies were important, but also what was important are controlled experiments that led to increased knowledge. So there was many scientists that conducted experiments which led to our increasing grasp of biology. And this all in turn led to the development of the cell theory, which is the most fundamental aspect of biology. You have to know it no matter what. It'll be on every single one of your tests. I'll be talking about cell theory in a bit. Damn. So, where does life come from? It was first described by Aristotle. This guy was a tryhard way back in the 1800s who described and tried to do everything. But they call him a philosopher. I don't even know why. He used his creative thinking and blah 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 and logic to explain the appearance of maggots on rotting meat. So when meat was rotting when left alone in its environment, there was maggots forming on top, and people were wondering why. And this guy was the one that came up with this ratchet idea called spontaneous theory. So it was also known as abiogenesis. So let's understand what that means. He said that life forms were formed from non-living matter. So literally you could have anything and life would just arise from it. That's what his idea was about the origins of life. Any non-living matter and life forms could be formed on top of it. That was abiogenesis, or the spontaneous generation theory. Now, this was further questioned by Francesco Redi. He challenged the theory of spontaneous generation through a simple jar experiment. This is what he did. He was ready to challenge it. Francesco Redi. What a real nigga. So his first aspect was he had a control group. So he literally had rotting meat left inside a jar and it was left open. So over time, what happened was that bugs would come in, bugs would enter, bugs would leave, and the meat would rot and break down, and there would be formation of decomposers on the meat, like these little worms that you see here. But then what he did was in his experimental group, he covered the lid with some kind of wrap so that no animals could get in, no animals could get out, and the air in the external environments couldn't get in either, so the decomposers wouldn't develop. So he said, I didn't do anything to this meat, so then why the hell wasn't there random life forms being formed out of nowhere? So this is what he used to challenge the spontaneous theory experiment. However, what ended up happening was the um, the microscope led to the discovery of single-celled organisms, and that also helped debate over spontaneous theory. Many people believe that an active principle or life force in air created organisms. So there was some special component of air that was making life readily available everywhere, which is like the most ratchet idea ever. Then came this big guy, this big boy right here with the mustache. He challenged absolutely everything. His name was Louis Pasteur, and he subsequently challenged the new theory of spontaneous generation through a swan nest flask experiment. This is how he did it. Needham's experiment was essentially he took chicken broth, he boiled it, and he sealed it in a container and he put it in there. But however, there was a period where he allowed the air to enter prior to sealing it so that microorganisms still formed. So he concluded that there was a random life force present in the air that was allowing everything to form. But Louis Pasteur went ahead and challenged this. This is how he did it. So he put the boiled chicken broth in a flask once again, and this killed all the microorganisms present. Now, before he exposed it to air, he created the swan next um, figure that you can see, and he heated that once again. So all the microorganisms in the air were automatically killed, and anything in the chicken broth was also killed. So the only way from the air to enter was to go through this tube and enter back in. Now, what this proved was any microorganisms or bacteria that were present in the air, those little guys, once they would enter this tube, they would get stuck here. The friction, the contact between the air molecules and the neck of the flask would trap the bacteria in here so that only air was entering and only air was leaving. Any bacteria or microorganisms would get stuck on this little edge right here, like this guy, and they wouldn't be able to access the chicken broth. So what this showed was that there was absolutely no microorganisms being formed in the chicken broth, and that spontaneous generation was false because this neck was still exposed to the air, but there was no life form present in the air that was just generating life everywhere. It was specifically the microorganisms that we were observing. So the air had no special property that was making life, it was just the microorganisms. Then came another man. His name was Rudolf Virchow. He's obviously Russian. And he suggested the theory of biogenesis. Now, and to know what the theory of biogenesis is, so biogenesis is essentially just a big fancy word for life arising from pre-existing life. So if you have a life form here, all it'll do is it'll essentially split and create two more pieces of life. That's exactly what cells do. So this led to the development of one crucial point in cell theory, that all cells come from pre-existing cells. That was discovered by Rudolf Virchow. Now the microscope also had significant development. It all started off in the 14th century when grinding of a lens was developed. So that's what people were doing back then because they had all this time. They were grinding lenses and they used it to fashion lenses to improve eyesight, so essentially in glasses. But they also knew that this was involved in looking at things in a much larger scale, so smaller things could be viewed much more easily. That's what he first did, Zacharias Janssen. In 1595, he was believed to be the inventor of the compound microscope. That means there's compound microscope essentially just means that there are more than one lens present in the microscope. And he created a microscope with two lenses, and he first used it to magnify multiple things. Then came Robert Hooke, and he observed cork using a three-lens compound microscope. So he had three different lenses, and he observed cork that he derived from a tree. And this is what he saw. This was the cork that he saw, and he noticed that it looked like little rooms, all separate rooms. So he gave it a name. He named it cellulae, which is the Latin word for cell. So that's where our word cell originally derives from. Then came Anton van Leeuwenhoek in 1675, just 10 years later. He was observing pond water and plaque from his own teeth, and he noticed the living cells that were in motion in the plant water, and he called them nanomolecules using his single lens microscope. So he was the first one to see living cells in motion. Robert Hooke was the first one to observe the cork tree cells and name it cellulae, which means cell. and Zacchaeus Janssen was the first person to invent the compound light microscope. Then came Robert Brown, and he put forth the suggestion that the granular spot that we see in cells, the big circle, is the nucleus and it's the control hub of everything and it's important for cell functions. And this is observed right here, here, and here. Those are the stromata, but these are the nucleuses. All these little things that we see. Then came Matthias Schleiden. He's obviously a German guy. And he noted that all plant tissues observed were made of cells. So all the plant tissues everywhere had cells in them. And Robert Brown was the one who noticed that all plant tissues have nucleuses in them, which are probably essential for all cell function. Then came Theodor Schwann, and he believed that there must be similarities between plant cells and animal cells. And he connected these ideas, and they proposed that all plant cells and animal cells were composed of cells, and that cells were the basic unit of organism. So when Schwann discovered the nuclei in the animal cells and Brown had already discovered the nucleus in the plant cells, he connected the dots and he noted that there must be similarities. So all animal tissue has cells, all plant tissue has cells. We know that all living things must have cells, and therefore that gave birth to cell theory. Cell theory states, which is essentially composed by Schleiden and Schwann at the start, all organisms are composed of one or more cells. That's it. If an organism doesn't at least have one cell, it's not a living thing. So that's how we can distinguish between living things and non-living things based on how many cells there are present. Any organism with one cell is a unicellular organism, like most bacteria, fungi, and protists. Any organism with more than one cell is a multicellular organism, like animals, plants, and me. I'm a multicellular organism. The cell is the smallest unit of life. So it's the basic unit of all life, and it's the smallest level of living things present. Then Robert Virchow, who we spoke about earlier, discovered that all cells arise from pre-existing cells, and that was a third point of cell theory, that all cells come from other cells by cell division, which was further discovered, and cell division can be broken down to mitosis and meiosis. No, we're not going to stop here today. Grind, don't stop. So now here we're presented with the information on the light microscope. And one sec. So the light microscope was the earliest simple microscope, which is essentially a bunch of magnifying glasses and used visible light. Therefore, they were a type of light microscope. So your eye would be placed right up here, and there was two ocular lenses which would observe the specimen. So your eye would receive all the light here, which was constantly getting refracted. And the light which was emitted from the light source at the bottom would be condensed by this condenser lens, go through the specimen. Therefore, the specimen is being observed, hit the objective lens, hit the ocular lens, and get into your eye. Your eye would make one last flip, and that way you could observe the specimen at a much higher magnification. Since it involves light, that's a light microscope, and as we spoke about earlier, Janssen was the first one to create the compound light microscope, which is two or more lenses stacked on top of each other. A compound microscope that uses visible light is a compound light microscope. We're not idiots. I said it like 6 times. So now let's talk about microscope technologies. One type of technology is the fluorescence microscopy. Now it uses various fluorescent substances attached to specific molecules in tissues which glow different colors when subjected to ultraviolet light. Let's look at an example. This is called bright field illumination. So this fluorescent substance, which is attached to a certain molecule, made the cytoplasm go orange and the nucleus go like a pink. And since it glows different colors, we can easily distinguish the different parts. This is dark field illumination, so obviously hidden in a dark area, light purple and purple. This is the phase contrast illumination, so this can actually help you determine the different phases. And I will. Yeah, so when you observe specimens to different levels of light, the different wavelengths will allow the light to reflect off differently, and thus you can see the different parts. Now there's electron microscopes, and that'll be the first thing I start about in the next half of this lesson.